A smooth golden dome sits on a dark rock more than 3 kilometers below the surface of the Gulf of Alaska. The ROV light touches it from one side, and for a moment it looks less like an animal than an object left behind. It is round, pale gold, slightly torn near the base, and surrounded by the kind of black seafloor where almost anything can appear unfamiliar if the camera arrives at the right angle. In the control room, scientists do what scientists often do when the deep sea offers something that does not fit cleanly into a category. They describe what they can see. A dome, a smooth surface, a small opening, a biological texture, something attached to rock. It might be an egg case. It might be a sponge. It might be the remnant of something that has already moved on. Nobody can be sure while the object is still sitting under pressure in the beam of a machine. The strange thing about deep-sea eggs is that they are not always easy to recognize as eggs. Some are wrapped in leathery cases that look like dark packets caught on stone. Some are pale capsules fixed to coral branches. Some are carried in the arms of mothers that will not feed again. Some drift in transparent sheets through water that seems empty until the camera gets close enough to see the small bodies inside. And some objects that look as if they might be eggs turn out not to be eggs at all. The golden orb that drew so much attention after the Alaska dive was later identified as part of the base of a giant deep-sea anemone, Relicanthus daphnae. It was not a developing animal sealed inside a shell. It was a remnant, a structure used by an animal to attach itself to rock. But the mistake was reasonable, because in the deep sea the first shape is rarely the final answer. If these quiet descents into the hidden ocean are what you came for, the next one will not be far away. This is where the story begins, not with one single egg, but with the difficulty of knowing what an egg even looks like in a world without sunlight, without warmth in the ordinary sense, without seasons that can be read from a shoreline, and without the easy access that lets a biologist return every morning to check a nest. At the surface, eggs can be seen in tide pools, tucked under kelp, glued to rocks, buried in sand, guarded under fins, or released in drifting clouds. In the deep sea, the same basic problem exists, but the conditions are harder. An embryo still needs oxygen. It still needs protection. It still needs time. It still needs enough stored energy to build a body before it can feed. But it must do all of this in cold water, under pressure, in darkness, and often far from any stable supply of food. Pressure itself is not a crushing force for animals that live there. Their tissues are mostly water, and water does not compress easily. The more difficult problem is chemistry, temperature, oxygen, predation, and time. Cold slows metabolism. Slow metabolism can help an embryo survive on limited energy, but it also makes development take longer. A shallow water egg may hatch in days or weeks. A deep sea egg can remain vulnerable for months or years. The camera does not always show this waiting. A dive may last a few hours. A ship may spend a few weeks in a region. A research program may return over several years if funding and weather and equipment allow it. But the egg has its own schedule. It is not moving at the speed of the expedition. It is moving at the speed of cells dividing in cold water. That is why a small object on the seafloor can contain a story much longer than the moment when it appears on a monitor. A pale capsule on a coral branch may have been there before the ship left port. A skate egg case near a vent may have been laid years before the ROV arm touched the bottom. A mother octopus in a rock crevice may have already spent months breathing water across her eggs before a human eye ever saw her. Eggs are simple only from far away. They are containers, but also compromises. Too many small eggs can scatter risk across thousands of offspring, but each embryo receives little protection and little stored energy. Fewer large eggs give each young animal a better start, but every loss matters more. A capsule can shield an embryo, but it must also let oxygen through. A mother can guard a brood, but she spends energy and may expose herself to predators. In the deep sea, many of these compromises become extreme. Food is limited in much of the water column and across wide areas of the abyss. The slow fall of marine snow carries fragments of the surface world downward, but it is thin by the time it reaches great depth. A large carcass can create a temporary feast. A hydrothermal vent or cold seep can support dense life through chemistry instead of sunlight, but for most animals, every reproductive decision is made under the pressure of scarcity. This makes eggs valuable in a way that is almost visible. They are food to many animals before they are offspring to one. A snail can rasp at them. A crab can tear at them. A fish can nose into the soft edges. Bacteria can colonize them. Sediment can cover them. Oxygen can fail to reach them if the water is still or if the capsule is buried too deeply. Even before hatching, the young animal is already part of the food web. The deep sea is often filmed as empty space. A light moves through black water, and only a few animals appear. But reproduction reveals that the emptiness is misleading. Somewhere in that volume, animals are mating, attaching, brooding, releasing, guarding, drifting, dying, and beginning again. The first strange eggs many people know by name are not deep-sea eggs at all, but they prepare the eye for the ones below. Mermaids' purses, the egg cases of skates, some sharks, and chimeras can wash up on beaches after the young animal has hatched. They are leathery, dark, and often rectangular or horned. Some have tendrils that wrap around seaweed. Some have corners that help them lodge in a stable place. They look like objects, not like the beginning of a fish. In deeper water, similar cases can be much harder to find. They may be attached to rocks, tucked among corals, laid among lava formations, or scattered across sediment where a trawl might damage them before anyone understands what has been collected. Unlike a bird's nest, they do not announce themselves with movement or sound. They wait in dim water, often at depths where a person will never go without a machine. One of the most remarkable examples was found near the Galapagos. In 2015, researchers using ROV Hercules were exploring volcanic seafloor north of the islands in a region marked by active hydrothermal vents. The ROV had cameras, lights, a robotic arm, and the slow, deliberate movement of a machine built for terrain that human hands cannot reach. When the camera panned across the lava, the seafloor was not just rock. It was marked by egg cases. They belonged to the Pacific white skate, Batharaja spinosissima, a cartilaginous fish related to rays and sharks. The cases were palm-sized, horned, and laid in numbers near active venting. More than 100 were observed in the area, and some were collected so that Dina could confirm the species. The pattern was not random. Many were positioned near black smoker chimneys, where hot, mineral-rich fluid rises from the crust. At first, this seems almost contradictory. Hydrothermal vents are harsh places. The fluid can be superheated before it mixes with seawater. Chemistry changes quickly across small distances. Metals and sulfides precipitate into black plumes. Animals that live directly in these systems are often highly specialized, yet here were the eggs of an animal that spends most of its life elsewhere, placed close enough to use the heat without being destroyed by it. The interpretation is cautious but powerful. Researchers propose that the skates were using the warmth of the vent field as a natural incubator. Deep-sea skates can have very long developmental times, in some cases estimated at more than 4 years. A slightly warmer environment can accelerate embryo development, shortening the time each egg remains exposed to predation, disease, and physical damage. A 4-year egg is difficult to imagine from a human perspective. It is longer than many research grants, longer than the working life of some pieces of equipment in corrosive seawater, longer than the time between many expeditions to the same remote site. A skate embryo developing inside a leathery case can spend years in darkness before it cuts its way out into a world it has never seen. The vent does not make the deep sea easy. It changes one variable. Warmer water can speed metabolism, but too much heat is fatal. Vent chemistry can support dense communities, but it can also be toxic outside the narrow tolerances of adapted organisms. The egg case must be placed in the right place, not simply near heat, but near a gradient where warmth is useful and conditions remain survivable. That is what makes the image so strange. Not just the cases themselves, not just their shape, but their position. Dark purses lying among volcanic rock, a black smoker in the distance. The ROV arm moving in carefully, not as a hunter, but as a temporary visitor trying to collect one piece of evidence without destroying the scene around it. The eggs suggest memory, or at least repeated behavior. When several layers of cases appear in the same area, it implies that adults have returned to that place again and again. The deep sea is not an empty floor where animals wander without pattern. It is mapped by temperature, current, chemistry, smell, slope, texture, and history. To a skate, a vent field may be not only a dangerous place, but a nursery. The Pacific white skate is one of the deeper living known skates. It has been documented across a wide range in the eastern Pacific, from the Galapagos region to waters farther north. Like other skates, it reproduces by laying eggs rather than giving live birth. The embryo develops inside the capsule, nourished by yolk, while seawater moves through openings and across the case so oxygen can reach the animal inside. This is a very different strategy from releasing millions of eggs into open water and leaving chance to do the rest. A skate egg case is an investment. It is a protected chamber built from collagen-rich material, tough enough to resist abrasion and some predators, shaped by evolution into a small structure that holds one developing fish for a long time. It is not delicate in the way a bird egg is delicate, but it is still vulnerable. A predator does not have to understand it as an egg. It only has to learn that something inside can be eaten. A crab can test the edges. A fish can bite. Microbes can enter if the case is damaged. Sediment can settle. If the egg remains too long, the chances of something going wrong accumulate. In the cold deep sea, time protects by slowing demand, but it also exposes by extending risk. The vent nursery is one answer to that problem. Not a perfect answer, not a safe place in any simple sense, but a place where the earth itself releases heat, and where a developing animal may borrow that heat long enough to hatch sooner. This is one of the quiet surprises of the deep ocean. The same systems that build sulfide chimneys and feed tube worms and shrimp can also influence the life history of an animal that does not belong entirely to the vent community. Reproduction crosses boundaries that adult bodies may not. An egg can make use of a place the adult only visits to leave it behind. There are other eggs in the deep sea that do not use hot vents but still depend on careful placement. Cirrate octopods, the group that includes many animals commonly called Dumbo octopuses, can attach large individual eggs to deep-sea corals and other bottom structures. These eggs are not broadcast as clouds. They are placed one by one, sometimes on living cnidarian colonies, and the tough capsule must protect the embryo while allowing development to continue in cold water. In 2017, NOAA's ROV Deep Discoverer recorded several observations of cirrate octopod eggs in the central Pacific. They were attached to octocorals, including colonies often called bubblegum corals. Some appeared ruptured at one end, not because they had been torn apart by a predator, but because the internal chorion had swollen during development and burst the outer capsule. This may help the young octopod escape when it is ready to hatch. It is a small detail, but it matters. Without seeing the egg in place, the swollen structure might be misread. A damaged capsule could look like a failed egg. A ruptured coating could look like predation. The ROV footage allowed scientists to connect shape with process. The embryo grows, the chorion expands, the tough outer case opens. What looks broken may be part of hatching. Cirrate octopods are not the same as the shallow water octopuses most people picture. Many have fins near the mantle and delicate webbing between the arms. They live in deep water, often moving slowly above the bottom or through the water column, using fins and arm webs in ways that suit a dark environment where speed is costly. Their eggs are often large compared with the eggs of shallow species. Large eggs are another answer to the deep sea. When the young hatch into a world where food may be scarce and encounters unpredictable, a larger, better developed hatchling may have an advantage. It can swim, sense, and feed more effectively. But producing large eggs means producing fewer eggs. The parent cannot fill the water with millions of chances. It places more of its future into each capsule. On the monitor, a single cirrate egg attached to coral does not look like a grand event. It looks like a small brown or pale object fixed to a branch in the black. But it links the reproductive life of one animal to the slow growth of another. Deep-sea corals may live for decades or centuries. Their branches create structure in a habitat where flat sediment can dominate for vast distances. For an egg, a coral is elevation, stability, and exposure to moving water. The egg needs flow. Oxygen must reach it, and waste must leave. In still water, a capsule can become its own trap. A coral branch lifted into the current offers a better position than sediment that can bury or smother the egg. This is why the shape of the seafloor matters. A canyon wall, a rocky outcrop, a coral garden, a vent field, or a seamount slope can all create nursery opportunities that a flat plain does not. Deep sea eggs often reveal the importance of surfaces. The animals themselves may be mobile, but their offspring begin attached to something. Rock, coral, sponge, lava, tube, mud. A mother chooses, or appears to choose, a place where the next stage can endure. The placement is a record of behavior that would be almost impossible to infer from a dead specimen alone. This is one reason ROV footage has changed the study of deep-sea reproduction. Nets can bring up animals, but they often destroy context. A trawl may collect an adult, an egg case, and a fragment of coral, but it may not show whether the egg was attached to the coral or merely tangled with it afterward. Video preserves relationships. It shows distance, orientation, depth, temperature, substrate, neighbors, and sometimes behavior. A specimen in a jar can answer some questions. A living animal in the light can answer different ones. The deep sea requires both. The camera sees the moment. The lab sees the tissue. DNA, microscopy, and anatomy can correct what the eye guessed too quickly. The golden orb is a perfect reminder of that. The first impression was not enough. Real eggs can also be difficult to recognize when they are carried instead of laid. In shallow seas, octopus mothers guarding eggs in dens are familiar to many biologists. The female attaches strands or clusters of eggs to a sheltered surface, cleans them, moves water over them, protects them, and usually dies after they hatch. In the deep sea, the same basic commitment can become an extraordinary test of endurance. At Davidson Seamount, off Central California, thousands of octopuses gather in a nursery known as the Octopus Garden. The site lies near the base of an extinct underwater volcano, about 3,200 meters below the surface, on a small hill where warm fluid seeps through cracks in the rock. From a distance, the nesting octopuses look like pale pearls scattered across the seafloor. The species is Musoctopus robustus, often called the pearl octopus by researchers because of its appearance in the ROV lights. Observations over several years showed that the site is used for reproduction. Adult males and females are present. Eggs are present. Hatchlings are present. Intermediate-sized animals are not. Feeding was not observed. The octopuses gather there to mate, nest, and die. That final part is not decoration. It is part of the biology. Like most cephalopods, these octopuses reproduce once and then die. The mother remains with her eggs, tending them while her own body declines. She does not leave to hunt. She does not rebuild herself for another brood. Her remaining life is transferred into care, oxygen, protection, and position. In the cold abyss, researchers expected these eggs might take 5 to 8 years, or perhaps longer, to hatch if they developed at ordinary deep-sea temperatures. At the Octopus Garden, the warmth from low-temperature hydrothermal springs appears to shorten that time dramatically. Using ROV imagery to follow individual mothers and broods, scientists found that eggs hatched in less than 2 years. Less than 2 years is still a long time for a mother to remain in one place without feeding. But compared with 5 or more years, it is a major reduction in risk. Every month saved is a month with fewer chances for predators to find the eggs, for the mother to fail, for sediment to shift, or for some other disturbance to interrupt development. Warmth does not remove danger. It narrows the window. The footage has a stillness that makes the behavior feel almost simple. A female octopus is tucked against rock. Her arms cover pale, sausage-shaped eggs. Small snails move nearby. Other octopuses occupy neighboring crevices. The ROV hovers just outside the nursery, a machine with thrusters and lights watching animals that have solved a reproductive problem with water slightly warmer than the darkness around them. The octopus garden is not only a nursery, it is also a food source for other animals. Eggs that fail, hatchlings that do not escape, and adult octopuses that die after reproduction become part of the surrounding community. Scavengers gather. Invertebrates feed. The dead bodies of mothers become energy for other lives. Reproduction and death are not separate scenes in the deep sea. They are often layered on the same patch of rock. A nursery attracts attention. It concentrates vulnerable bodies in one place. Predators and scavengers do not need to search the entire seamount if the reproductive site is predictable. The advantage of warmth must be balanced against the danger of gathering, yet the number of octopuses suggests that, over time, the balance works. The site persists because enough offspring survive to make the journey worthwhile. Researchers counted more than 6,000 octopuses in part of the site and estimated that 20,000 or more could be using the nursery. That is not a random crowd. It is one of the largest known gatherings of octopuses on the planet, assembled in darkness around small leaks of warmth from the Earth. This does not mean every deep-sea octopus uses thermal springs. It means that in this place, for this species, under these conditions, a geological feature has become part of reproductive strategy. The same ocean can produce many answers. Some octopuses attach eggs to corals, some guard them in dens, some may use warmth when it is available. Many species remain poorly observed, and their reproductive lives are still known from scattered moments. The deep sea often teaches by exceptions, and ROV finds something unusual, and the unusual thing becomes a question. Is this rare because it barely happens, or rare because humans barely see it? The distinction matters. A behavior observed once may be an accident, or it may be common in places our cameras almost never visit. Squid add another layer to this problem. For a long time, many biologists assumed that squid generally laid eggs and left them, either attaching them to the seafloor or releasing gelatinous masses into open water. This made sense from what could be observed in shallow species and from what specimens revealed. But the deep sea has repeatedly shown that reproductive behavior can be more varied than expected. In 2002, Umbari's ROV Tiburon recorded a female Gonatus onyx squid carrying a large mass of eggs between her arms at depths of roughly 1,500 to 2,500 meters. The egg mass was not attached to the bottom. It moved with her. It was suspended from hooks on her arms, and as she swam, she appeared to pump water through the cluster, helping the embryos receive fresh oxygenated water. The behavior was later described as post-spawning egg care. Female Gonatus onyx can carry clusters of up to about 3,000 eggs. During brooding, they cannot move quickly. They are thought to be more vulnerable to deep-diving predators such as whales and elephant seals. The mother also does not feed while carrying the eggs. The strategy is costly, but it protects the embryos in a way drifting eggs or abandoned capsules cannot. Imagine the same animal without the romance of human language. A squid in cold black water, arms occupied, body aging, tentacles gone or reduced after spawning, moving slowly because speed would tear or abandon what she carries. She is not guarding a nest. She is the nest. The nursery is not a place on the seafloor. It is suspended from her body. This changes the meaning of open water. The midwater can seem emptier than the bottom because there is no terrain, no wall, no rock, no chimney, no skeleton, no branch. But eggs can exist there in mobile form. A brooding squid turns the water column into a moving nursery, carrying the next generation through depth layers where currents, predators, oxygen, and temperature all change. The observation did not mean that all squid brood. Most do not, or at least have not been observed doing so. It meant that one deep-sea squid did something scientists had not expected from squid as a group. Since then, brooding has been documented in only a handful of deep-sea and open ocean squid species, including Gonatus and Bathyteuthis. Each observation remains valuable because it is so difficult to see the behavior directly. In Monterey Bay, Mbari researchers later recorded a Bathyteuthis squid carrying hundreds of eggs in a gelatinous sheet. The sheet held roughly 360 developing offspring, each embryo in its own capsule. The animal was seen at about 1,390 meters depth, far offshore, in a place where the water column itself is the habitat. The eggs looked like small, pale points embedded in a transparent or iridescent structure, held in the mother's arms. That image is different from a mermaid's purse on lava or an octopus brood under rock. It is more fragile-looking, less anchored to the world. But it is not passive. The mother holds the sheet. Her body creates the moving boundary between embryos and the rest of the ocean. The eggs are carried through the dark by an animal that has given up almost everything else. In 2024, Mbari and collaborators reported another striking observation from Mexico's Gulf of California. During a 2015 expedition, Rev. Doc Ricketts encountered a squid in the family Gonatidae brooding exceptionally large eggs. The eggs were about 11.6 millimeters across, roughly twice the diameter of eggs previously seen in brooding Gonatus squids. The mother carried far fewer eggs, perhaps 30 to 40 rather than thousands. This is a familiar reproductive trade-off in a deep-sea form. Fewer eggs, larger eggs, more investment per embryo. A larger egg contains more yolk. More yolk can produce a larger hatchling. A larger hatchling may be better able to swim, hunt, and survive in a food-poor environment. But the mother has fewer chances. If a predator takes the brood, the loss is enormous. The Gulf of California squid may represent an undescribed species. That statement is careful because video alone is rarely enough for a final taxonomic decision, especially in deep-sea squid, where many species are known from damaged or incomplete specimens. Researchers reviewed the RUV footage and examined similar specimens from previous expeditions, but uncertainty remains part of the story. The camera sees the arms folded around the eggs. It sees the color, the size, the slow motion of the animal in dark water. It does not automatically provide a name. Names require comparison, anatomy, sometimes DNA, and often a specimen. The deep sea gives the image first, then makes the answer difficult. Still, the image is enough to show that deep-sea reproduction can be more intimate than expected. A squid is often imagined as a fast animal, all jets and arms and sudden motion. A brooding squid is almost the opposite. She is slowed by care. Her survival becomes less important than the water moving through the eggs she carries. This slowness has consequences. Predators can exploit it. A sperm whale or elephant seal diving through the mesopelagic and bathypelagic zones may find a brooding female easier to catch than a free-swimming squid. The eggs that protect her offspring also make her more vulnerable. In the deep sea, care is not sentimental. It is a risk written into movement. The same is true for octopus mothers that remain with eggs and do not feed. The same is true for skates that leave egg cases in a place where embryos will remain for years. Every strategy solves one problem by creating another. Protection requires cost. Warmth requires placement. Size requires fewer numbers. Attachment requires the right surface. Brooding requires vulnerability. This is why strange eggs are not only strange in shape, they are strange because each shape reveals a decision made by evolution under conditions humans experience only indirectly. A horned case near a vent, a pale cluster under an octopus arm, a gelatinous sheet in the arms of a squid, a capsule on coral. Each one is a different answer to the same question. The question is, how to begin life in a place that gives very little away. Not all deep-sea eggs belong to large or charismatic animals. Gastropods at hydrothermal vents produce egg capsules too. In the late 20th century, researchers retrieved egg capsules from prosobranch gastropods at the Galapagos Rift and Juan de Fuca Ridge vent fields. These were not dramatic objects in the popular sense. They were small reproductive structures from animals living in dense, chemically powered communities where the seafloor itself releases energy. Vent snails, limpets, and other mollusks face reproductive challenges that differ from those of animals on the abyssal plane. Their habitat can be rich, but patchy. A vent field may support dense life, but it is also unstable over geological and ecological time. Vents can weaken, shift, or stop. New vents can open elsewhere. Larvae must either remain near suitable habitat or disperse far enough to find another chemical island in the dark. A hydrothermal vent is not simply a hot spring with animals around it. It is a boundary between seawater and the interior of the Earth. Cold oxygenated water seeps into cracks, heats near magma or hot rock, reacts chemically, rises again, and emerges loaded with reduced compounds and dissolved minerals. Microbes use chemical energy to build organic matter, and animals feed directly or indirectly on that microbial production. For an egg, this environment is both opportunity and threat. Close to vent flow, temperature and chemistry can change sharply. A few centimeters can separate tolerable warmth from lethal exposure. Sulfide can feed microbial symbionts in adult animals, but it can also poison organisms that are not adapted to it. Oxygen may be abundant in surrounding seawater and depleted in certain microhabitats. Placement matters at the scale of a body length or less. This makes vent reproduction difficult to interpret from scattered samples. An egg capsule collected near a vent may have been laid there by an adult resident. It may have drifted or been moved. It may belong to a species whose larvae disperse widely. It may reveal a reproductive pattern or only a moment. To understand it, researchers need repeated observations, specimens, environmental measurements, and sometimes luck. The ROV pilot does not always know, while hovering over the bottom, whether the small pale clusters on a rock are eggs, bacteria, tissue, empty cases, or the damaged remains of some other animal. The manipulator arm can collect only a few things. Every sample changes the place it touches. Every observation is a compromise between learning and leaving the scene intact. Cold seeps create another kind of reproductive landscape. Instead of hot water rising from volcanic systems, cold seeps release methane, sulfide, or other reduced chemicals from sediments. They can support mussel beds, clam fields, bacterial mats, tube worms, crabs, and other animals. Like vents, they are patchy sources of energy in the deep sea. Like vents, they can concentrate life, and therefore concentrate feeding, mating, eggs, and larvae. In seep and vent habitats, the egg is part of a chemical geography. A larva hatching into the water must eventually find suitable conditions, or it will die in an ocean that is vast relative to its body. Some larvae can drift. Some may respond to chemical cues. Some may remain close to the bottom. For many species, the details remain incomplete because the early life stages are small, delicate, and hard to identify. The youngest animals are often the least visible. Adults may sit on rock, crawl over chimneys, wave-feeding structures in current, or appear under a camera beam. Eggs and larvae may be hidden, transparent, microscopic, or indistinguishable from debris unless collected and studied. The beginning of deep sea life is often smaller than the tools built to find it. This is why some of the most important discoveries are not dramatic in the moment. A tiny capsule on a coral branch, a cluster of eggs under an octopus, a sheet held by a squid, a dark leathery case among lava. They matter because they connect adult animals to the next generation. Without them, the deep sea becomes a museum of adults with no explanation for how the world renews itself. Reproduction also changes how we understand deep sea abundance. A camera may show only a few animals in a given frame, but that frame is a tiny slice of an enormous volume. The deep sea is sparse at small scales and immense at large scales. Eggs make this scale visible in a different way. A single mother may carry hundreds or thousands. A single nursery may contain thousands of adults. A single coral garden may hold many hidden capsules. The water column above the seafloor is not empty either. It contains layers of migrating animals, gelatinous predators, lanternfish, crustaceans, squid, siphonophores, jellies, and countless larvae. Each night, many animals rise toward shallower water to feed and descend again before daylight. This daily vertical migration is one of the largest movements of biomass on Earth, and eggs and larvae are embedded in that moving system. Some eggs are buoyant. Some sink. Some drift inside gelatinous masses. Some are attached. Some are guarded. The fate of each depends on density, currents, predators, temperature, oxygen, and developmental time. In the open ocean, an egg mass can be enormous yet nearly invisible because it is transparent and surrounded by water. Divers occasionally encounter giant gelatinous squid egg masses in shallower water, but many such structures may form deeper and pass unnoticed. The deep sea resists easy categories here too. An egg mass found drifting at one depth may have been produced somewhere else. It may rise or sink as the embryos develop. It may be torn apart by currents or animals before anyone sees it. A dead or empty mass may be easier to find than a fresh one. The evidence often arrives after the main event has already happened. A camera beam catches what remains. A torn sheet. A capsule with a hole. A case without an embryo. A mother near the end of her life. A new hatchling leaving rock. In a place where direct observation is rare, remains are not secondary. They are often the only doorway into behavior. This can make the deep sea feel like a place of endings, because so much evidence comes from bodies, fragments, shells, scars, beaks, and empty cases. But an empty egg case is not only a sign of loss, it may also be a sign that something escaped. The small opening that looks like damage may be the route by which the young animal entered the world. Hatching itself can be physically demanding. An embryo must break, cut, dissolve, or push through the protective structure that kept it alive. In skate egg cases, the developing animal eventually emerges from the capsule as a small skate, already shaped for life on or near the seafloor. In cephalopods, hatching may involve swelling, rupture, or escape from membranes. The egg must be strong until it must fail. That is one of the most elegant tensions in any egg. It must protect without imprisoning. It must keep predators and microbes out while letting oxygen in. It must remain intact through days, months, or years, and then open at the right time for an animal too young to have any experience of the world outside. In cold water, oxygen can be relatively abundant because cold water holds more dissolved gas, but oxygen availability is not uniform. Oxygen minimum zones occur where respiration consumes oxygen faster than circulation replaces it. Some deep-sea animals are adapted to low oxygen, but embryos may have different tolerances from adults. An egg laid in the wrong microhabitat may suffocate even if the adult could survive nearby. Temperature is equally complex. Warmer water can speed development, as seen in the skate egg cases near vents and in the octopus nursery at Davidson Seamount. But faster is not always better if it pushes metabolism beyond oxygen supply or developmental limits. Too cold, and the embryo remains vulnerable for too long. Too warm, and development may fail. The useful range can be narrow. The deep sea does not offer many gentle solutions. It offers gradients. A little more oxygen here. A little more current there. A surface raised above sediment. A crack with warmth. A coral branch in flow. An animal's arms. A tough case. A transparent sheet. Life begins by finding the least dangerous version of an impossible place. The abyssal plains, stretching across huge areas of the planet, may seem like the hardest place for eggs to matter because there is so little structure. Sediment covers everything. Food arrives as a thin rain from above. Animals are often small, slow, widely spaced, and tuned to scarcity. Yet even there, reproduction continues. Burrowing animals release gametes, snails lay capsules, fish find mates. Invertebrates produce larvae that rise into the water or remain near the bottom. Much of this is known imperfectly. The abyss is vast, and sampling it is difficult. A box core can bring up sediment and small animals. A trawl can collect larger organisms but damages many soft-bodied forms. A lander can watch one patch for a time. An ROV can follow a transect. None of these tools sees the whole life cycle of most species. Eggs are among the easiest stages to miss. A small capsule buried partly in silt may pass beneath the camera without anyone noticing. A gelatinous egg mass can drift just outside the light. A brooding animal can avoid the vehicle or be mistaken for an ordinary adult if the eggs are hidden beneath the arms. The deep sea does not hide things deliberately. It hides them by scale. Scale is the constant obstacle. The ocean is deep, wide, moving, and three-dimensional. A ship on the surface lowers a machine into a volume that dwarfs it. The cable pays out kilometer after kilometer. The ROV reaches the bottom, and its lights reveal only a cone of visibility. Beyond that cone, the same habitat continues, unseen. There may be eggs 10 meters away, or 10 kilometers away, or not at all. This is why repeated observation is so important. A single egg case near a vent might be coincidence. Many cases in layers near the hottest vents become evidence of a pattern. A single octopus with eggs might be a curiosity. Thousands of nesting octopuses, monitored over years, become a nursery. A single brooding squid might overturn an assumption. 17 observations over decades suggest a behavior that, while still rare to us, is real enough to study. Deep sea eggs also change how we think about time after death. The golden orb was not an egg, but it looked like one because it was a biological remnant with a rounded shape and a small opening. It had to be collected, examined, and sequenced before its identity became clearer. The same caution applies to actual egg cases. Empty, damaged, or weathered structures can persist after the animal has left or died. A case without contents is still data. It can reveal where eggs were laid. It can sometimes reveal species through shape or DNA. It can show repeated use of a site, but it can also mislead if treated as fresh evidence. Was the egg viable? Did it hatch? Was it eaten? Was it transported? How long has it been there? These questions are not always answerable from appearance alone. The deep sea is full of objects that have outlived their moment. Worm tubes after worms are gone. Coral skeletons after polyps die. Whale bones after flesh is eaten. Egg cases after young have hatched or failed. The seafloor remembers life in fragments, and those fragments can be read only carefully. There is no shame in a first guess being wrong. The danger is refusing to let the evidence change it. The golden orb became famous partly because it invited imagination, but its real identity is more interesting than the speculation. It belonged to an animal, a giant anemone-like creature of the deep, and it showed that even attachment structures can appear mysterious when found without the body that made them. Relicanthus daphne is a large deep-sea cnidarian, often described as an anemone ally. It can have long, pale tentacles and lives attached to hard surfaces in deep water. The golden material appears to be part of a cuticle or base associated with anchoring the animal. Microscopy and genetic analysis linked the specimen to this animal rather than to an egg case, sponge, or unknown organism. That correction matters for a script about eggs. because it shows how easily human interpretation can run ahead of evidence. In the deep sea, shape alone is not enough. A smooth dome may be tissue. A torn opening may not be a hatch. A cluster may not be a brood. A capsule may not be empty. Every claim needs the support of context, collection, comparison, and sometimes years of analysis. But the question that first surrounded the orb was still a good question. Could this be an egg? That question belongs in the deep sea, because eggs there can look unlike the eggs we know. They can be tough, dark, gelatinous, hidden, mobile, scattered, singular, or attached to animals that are themselves barely familiar. A deep-sea egg is not just a shape, it is a strategy made visible. Consider the difference between a broadcast spawner and a brooder. In broadcast spawning, adults release eggs and sperm into the water, and fertilization happens externally. This can produce huge numbers of embryos, but most will die. It works best when timing, currents, population density, and environmental conditions allow enough gametes to meet and enough larvae to survive. It is a strategy of abundance and loss. Brooding is almost the opposite. The parent retains some control after fertilization. It may hold eggs, guard them, clean them, ventilate them, or place them carefully. Brooding reduces some risks but increases others. The parent is tied to the embryos. It may be unable to feed or flee. It invests time and energy into fewer offspring. In deep water, where development can be slow, that investment can dominate the end of the parent's life. The mother octopus at the octopus garden does not have a second career after the eggs hatch. The brooding squid does not resume ordinary hunting while carrying her mass. These animals are not symbols of sacrifice in the human sense. They are organisms following life histories shaped by selection. But from the outside, the result is stark. Reproduction consumes them. That is one reason the footage can feel quiet rather than spectacular. Nothing attacks. Nothing explodes into motion. A pale octopus holds position. A squid drifts with arms closed. A skate egg case sits on lava. The drama is biological, not theatrical. The event is slow enough that a human viewer has to learn how to see it. Many deep-sea embryos hatch into larvae that do not look like the adults. Some enter the plankton, feeding or drifting before settling. Others hatch as more developed juveniles with fewer planktonic stages. The balance depends on species, egg size, yolk supply, habitat, and evolutionary history. A larva that disperses widely may find new habitat, but it may also be lost. A juvenile that stays near the bottom may avoid some risks, but depend on local conditions. Dispersal is especially important for patchy habitats, like vents and seeps. A vent field is not continuous across the ocean. It is a point in a network. Larvae must move between points through water that may not support the adults at all. How they find suitable habitat remains one of the major questions in deep-sea ecology. Chemical cues, currents, larval behavior, and chance all appear to play roles. A larva from a vent snail or tubeworm may begin life in a place where the chemistry of the earth feeds a dense community, then drift through ordinary deep water where that chemistry is absent, then settle only if it encounters another rare patch of the right conditions. The ocean between nurseries is not a road. It is a vast moving field of probabilities. Eggs are the first part of that probability made visible. They are the point where the adult body has done what it can, and the environment takes over. Predation on eggs is not an accident at the margins of the story. It is part of the reproductive equation. At the octopus garden, dead adults and vulnerable broods support scavengers and other invertebrates. At skate nurseries, egg cases may attract animals able to feed on embryos or empty cases. In the midwater, brooding squid may be targeted by predators because their movement is limited. Eggs are offspring, but they are also packets of nutrition. In food-poor habitats, a packet of yolk is not ignored. The same stored energy that gives an embryo a chance also makes it worth eating. The capsule, the mother's arms, the rock crevice, the coral branch, and the vent warmth all exist partly because other mouths exist. Reproduction is never separate from hunger. This is why egg protection can evolve in so many forms. Tough collagen cases in skates, gelatinous sheets in squid, den guarding in octopuses, coral attachment in cerates, capsules in snails. Each structure changes the odds. None guarantees success. A successful strategy is not one where every offspring lives. It is one where enough survive over generations for the lineage to continue. That may sound cold, but it is also what makes the deep sea feel real. These animals are not trying to be strange. They are not performing mystery for the camera. They are solving ordinary problems under extraordinary conditions. The shapes look strange because the conditions are strange to us. The more closely we look, the less the deep sea behaves like a single place. The eggs of the midnight zone are not the eggs of the abyssal plain. The eggs near vents are not the eggs of open midwater. The eggs attached to coral do not face the same risks as eggs buried in sediment. Depth is only one variable. Habitat is the rest of the story. The twilight zone, from roughly 200 to 1,000 meters, still receives faint traces of sunlight in its upper reaches, though not enough for photosynthesis through most of it. Below that, in the midnight zone and bathypelagic depths, sunlight is gone. Animals rely on food from above, predation, scavenging, chemosynthesis in special habitats, and bioluminescent signals. Reproduction in these layers must work without visual cues that surface animals might use. Many animals still use light, but it is light made by bodies. Bioluminescence can attract mates, confuse predators, lure prey, or help animals remain camouflaged against downwelling light. Eggs themselves are not usually the glowing stars of this system, but the reproductive lives of adults may depend on finding each other in a huge dark volume. Before there can be an egg, there must be an encounter. Finding a mate in the deep sea can be one of the hardest problems of all. Population densities may be low. Individuals may be scattered across depth layers or seafloor features. Currents move odors and signals. Some species solve this with strong chemical cues. Some gather at predictable sites. Some may synchronize with environmental conditions. Some may simply produce enough gametes that rare encounters still lead to reproduction. A nursery is therefore more than a place where eggs are laid. It is evidence that adults found a way to gather. At Davidson's Seamount, that gathering is visible by the thousands. At the Galapagos vent field, it is visible in layers of skate egg cases. In the midwater, a gathering may pass unseen because there is no landmark, only animals meeting in a moving depth range. The deep sea holds both kinds of reproduction, the anchored and the drifting, the crowded and the solitary. The tools that reveal these scenes are part of the story. ROVs carry cameras, lights, thrusters, manipulator arms, suction samplers, temperature probes, and sometimes high-definition or 4K imaging systems. Submersibles carry people inside pressure spheres. Autonomous vehicles map terrain with sonar. Landers sit on the seafloor and watch. Nets collect specimens, though often damaged. Museum jars preserve bodies for comparison decades later. Each tool sees differently. A net can sample animals that avoid cameras, but it loses behavior. A camera preserves behavior, but only where it looks. A suction sampler can collect delicate material, but only if the pilot can reach it. Dina can identify tissue, but it may not explain what the animal was doing. A long-time series can reveal development, but only if the instrument survives and returns data. This is why deep-sea egg discoveries often move slowly from curiosity to explanation. The golden orb required collection and lab analysis. The octopus garden required repeated visits and monitoring over years. The skate egg cases required video surveys, specimens, and Dina confirmation. The brooding squid observations required careful review of footage and comparison with known specimens. The answer is rarely immediate. That delay is part of the atmosphere of real exploration. Not every object becomes a revelation on the day it is found. Some sit in a lab. Some are compared with museum specimens. Some wait for a specialist. Some remain uncertain. The deep sea does not resolve itself on a video's schedule, yet the moment of discovery still matters. A control room is dark, except for screens. The ship rolls slightly above kilometers of water. Pilots move the ROV with small corrections. Scientists watch the feed, speaking carefully because every claim is being made in public or recorded for later. Then, something appears, and the room changes. Not loudly, necessarily, sometimes just with a pause. A pale object in the beam, a case among rocks, a mother with arms closed, a coral branch carrying a capsule. The first human reaction is often not certainty, but attention. Attention is the beginning of science in these places. Without it, a skate nursery might be mapped as ordinary vent terrain. An octopus aggregation might be treated as a strange crowd without understanding its reproductive purpose. A squid with eggs might be missed as a malformed silhouette. A serrate egg on coral might be dismissed as debris. The deep sea rewards slow looking. Slow looking also prevents exaggeration. Not every strange egg is from an unknown species. Not every unknown object is an egg. Not every nursery is unique. Not every behavior observed once is typical. The most trustworthy deep-sea stories leave room for uncertainty. They say what was seen, what was measured, what was collected, what is inferred, and what remains unresolved. This is especially important with reproductive behavior because the stakes of interpretation are high. If a species depends on a particular nursery, that habitat may be more important than its size suggests. If eggs take years to develop, disturbance can affect generations before anyone sees population decline. If adults gather in predictable locations, they may be vulnerable to human activity in ways a broad distribution would conceal. Deep sea mining, bottom trawling, cable laying, pollution, warming, and deoxygenation can all intersect with reproductive habitats. A vent field is not just mineral deposit and chemistry. It may be an incubator. A coral garden is not just structure. It may hold eggs. A seamount is not just a feature rising from the seafloor. It may concentrate mating, feeding, brooding, and dispersal. Damage to adult habitat is visible when corals break or animals disappear. Damage to eggs can be quieter. A trawl may remove cases before they hatch. Sediment plumes may smother capsules. Noise and disturbance may alter adult behavior. Warming can change developmental timing. Oxygen loss can squeeze the depth ranges where embryos can survive. The consequences may appear years later, when fewer young animals enter the population. The deep sea is often managed with incomplete maps. Many areas have not been visually surveyed in detail. Even when bathymetry is known, the biological communities may not be. A site can be licensed, fished, crossed, or disturbed before anyone knows whether it contains nurseries. Strange eggs are therefore not only curiosities, they can be indicators of places that matter disproportionately to future life. An egg case is small, but it can change the meaning of a landscape. A lava field becomes a nursery. A warm crack becomes a developmental shortcut. A coral branch becomes a cradle. A mother's arms become a shelter. A transparent sheet becomes a moving room in the dark. Once eggs are seen, the deep sea is no longer just a place where adult animals live. It is a place where they begin. Deep sea fish use many reproductive strategies beyond skates. Some produce pelagic eggs that drift upward or remain in the water column. Some guard nests. Some attach eggs to substrates. Some are live-bearing. The diversity is enormous, and many species are poorly known. In some cases, scientists can infer reproduction from gonads in specimens, larvae in plankton samples, or seasonal patterns in captures. Direct observations remain rare. Rare does not mean unimportant. It often means underobserved. A camera may record a behavior for the first time long after the behavior evolved. The first observation of a brooding deep-sea squid did not create the behavior. It revealed that a long-standing assumption was incomplete. The same may happen with other animals whose eggs have not yet been seen in place. Somewhere beyond the current reach of cameras, there may be nurseries on canyon walls, egg cases in sponge fields, capsules under ledges, brooding animals in oxygen-poor water, larvae rising from seeps, and gelatinous masses drifting through layers no vehicle has crossed at the right moment. The size of the unknown should not be exaggerated into emptiness. The deep sea is studied, sampled, mapped in places, and increasingly observed with sophisticated tools. But it remains too large for confidence in every detail. The honest statement is not that we know nothing. It is that what we know is uneven, bright in some places and dark in others. Eggs often sit in those darker parts of knowledge because they require timing. An adult can be present for years. A skeleton can remain. A coral can grow slowly in one place. An egg may be visible only during a narrow phase, or identifiable only when development reaches a certain stage, or present only in a breeding season that is not obvious from the surface. Some deep-sea animals may reproduce seasonally, linked indirectly to surface productivity. When phytoplankton blooms at the surface, more organic matter can sink, and the pulse of food can eventually reach deeper communities. Other animals may reproduce more continuously, especially in stable environments or chemically fueled habitats. The deep sea has seasons, but they can be delayed, muted, and difficult to read. Marine snow is one of the main connections between surface and depth. It is made of dead plankton, fecal pellets, mucus, molts, mineral grains, and other particles that fall through the water. By the time it reaches great depth, much of its energy has been consumed. But pulses still matter. A rain of food can support adults, scavengers, microbes, and perhaps reproductive timing in some species. An egg laid in the deep sea may therefore carry the surface inside it. The yolk was built from food the parent found, and that food may have begun as sunlight captured by plankton far above. Even in total darkness, the embryo can be connected to a bloom it will never see. At vents and seeps, the connection is different. The energy begins with chemistry rather than sunlight. Microbes use compounds such as hydrogen sulfide or methane, and animals feed on the microbes or host them as symbionts. Eggs laid in these systems may be part of a food web that does not depend directly on the daily fall of surface production. But larvae still move through the ocean, and many adults still interact with surrounding deep-sea communities. This means that deep-sea eggs can represent two great energy pathways on Earth. Some are built from sunlight that sank. Some are built from chemistry that rose. Both begin in darkness at the moment the camera finds them. The darkness itself is not empty or evil. It is simply the condition under which these systems operate. Animals do not experience it as a metaphor. They experience gradients of pressure, temperature, oxygen, flow, chemical concentration, vibration, smell, touch, and light produced by other animals. An egg does not need a sunrise. It needs the right exchange across its boundary. The boundary is everything: shell, capsule, jelly, case, skin, arm, rock crevice, coral surface, vent gradient. Each egg is protected by boundaries and threatened by them. Too open, and predators or microbes enter. Too closed, and oxygen fails. Too exposed, and it is eaten. Too hidden, and water does not move. Skate egg cases often have openings or structures that permit seawater exchange while protecting the embryo. Cephalopod egg capsules vary widely, but many involve membranes, jelly, or tough coatings. Gastropod capsules can hold embryos in protective structures attached to surfaces. The details differ, but the central problem is consistent. Development requires controlled contact with the environment. In human terms, it is tempting to imagine an egg as a sealed room. Biologically, it is more like a selective border. It must let certain molecules pass and resist others. It must manage water, oxygen, waste, mechanical stress, and biological attack. In the deep sea, where development can be slow, the border has to keep working for a long time. This is why dead or preserved eggs can be hard to interpret. A membrane that was flexible under pressure may collapse at the surface. A gelatinous structure may deform when collected. A case may dry, shrink, tear, or lose color. An animal removed from deep water may look grotesque compared with its living form. Deep sea bodies often change when brought into human conditions, and eggs are no exception. The most truthful image is often the one taken in place, before the pressure changes, before the temperature rises, before the soft tissue loses its shape. But the most complete answer often requires removing a sample. The deep sea asks scientists to choose between context and analysis. Then, try to rebuild the whole from both. When a suction sampler collected the Alaska golden orb, it changed the object from a seafloor mystery into a laboratory specimen. Without that collection, its identity might have remained speculation. With collection, the object left its habitat, but it entered the chain of evidence. The same is true for egg cases collected by ROV arms. The sample is a loss from the seafloor and a gain for knowledge. Ethical collecting in deep-sea research often means taking as little as possible and documenting as much as possible. Video, still images, coordinates, depth, temperature, associated animals, and substrate all matter. A specimen without context is less useful. Context without a specimen may remain uncertain. The strongest discoveries combine both. The skate eggs near Galapagos were not simply photographed and declared. Some were collected. DNA confirmed their species. The positions were analyzed relative to vent structures. The observation was compared with what is known about skate development. The conclusion emerged from several lines of evidence, not from surprise alone. That is the difference between mystery and science. Mystery begins when the object appears. Science begins when the object is described, tested, compared, and allowed to become something more specific than wonder. Wonder still remains. Knowing that a skate may use volcanic heat to speed its embryos does not make the image less strange. It makes it stranger, in a better way. The egg cases are no longer odd shapes near vents. They are part of an ancient reproductive problem solved with geological heat. Knowing that thousands of octopuses gather at warm springs does not remove the gravity of the scene. It deepens it. The pale bodies are not a random crowd. They are mothers, mates, dying adults, developing embryos, hatchlings, scavengers, and the energy of a nursery cycling through a cold seafloor community. Knowing that a squid carries eggs through the mid-water does not make her less haunting. It makes the slow movement meaningful. She is not simply drifting, she is ventilating a future she will not enter. The title, Strange Eggs Found in the Deep Sea, could suggest a collection of oddities. But the deeper story is not a cabinet of curiosities. It is a study of how life reproduces in habitats where humans rarely witness the beginning. The strangeness comes from adaptation, not from fantasy. Adaptation does not mean perfection. Many eggs fail. Many embryos are eaten. Many larvae never find habitat. Many mothers die without successful hatching. The deep sea is not a place where every rare strategy triumphs. It is a place where strategies are tested by cold, darkness, pressure, chemistry, currents, predators, and time. Failure is built into reproduction. An empty case may be successful or unsuccessful. A dead mother may have hatched her eggs or lost them. A damaged capsule may have opened naturally or been breached. A nursery may produce many young, but only a fraction may survive to adulthood. The camera can catch the evidence without knowing the outcome. Scientists try to close that gap by returning. The Octopus Garden study monitored individuals, broods, and conditions over multiple years. That kind of patience is rare but essential. It turns a beautiful scene into a life history estimate. It shows that eggs hatch in less than 2 years at the warm spring site, rather than remaining a vague symbol of maternal care. Long-term observation can also reveal what animals are not doing. At the octopus garden, researchers did not observe feeding, and they did not see intermediate-sized octopuses. That absence matters. It supports the interpretation that the site is used specifically for reproduction, not as a general habitat for all life stages. Absence is tricky evidence, but when combined with repeated observations, it can be informative. If many dives over time show adults, eggs, hatchlings, dead adults, and no feeding, the pattern becomes hard to ignore. Deep-sea science often works this way. It does not rely only on what appears. It also learns from what never appears despite careful watching. A nursery is defined as much by repetition as by abundance. In some ways, deep-sea eggs make the ocean feel older than the animals themselves. A skate lays an egg near heat from the crust. An octopus uses a seamount formed by volcanic processes. A coral branch that may have grown over many years holds the egg of an octopod. The time of geology, the time of growth, and the time of embryonic development overlap in one frame. Yet the animals are not fossils or relics. They are modern organisms living now, responding to present conditions. The Pacific white skate is not a leftover from a prehistoric world. The pearl octopus is not a symbol from deep time. The squid carrying eggs is not a monster from imagination. They are living animals with bodies shaped by ancestry and environments that still operate today. It is important to let them remain animals. A mother octopus breathes water across eggs because the embryos need oxygen. A squid holds an egg sheet because letting it drift may reduce survival. A skate leaves a case near warmth because development may be faster. These acts are strange to us, but ordinary to the life histories that produced them. The word strange should not become a way of making the animals unreal. It should be a way of admitting that our expectations were built at the surface. At the surface, temperature changes quickly with season and weather. Light defines day and night. Many eggs develop in comparatively accessible habitats. Humans can snorkel, dive, collect, revisit, and observe. In the deep sea, revisiting requires ships, crews, weather windows, fuel, navigation, robotics, pressure-rated systems, and a reason strong enough to justify the cost. This logistical barrier shapes knowledge. It is not that the deep sea is unknowable, it is that every observation is expensive in time and engineering. A simple question, such as how long an egg takes to hatch, may require years of footage and repeated identification of the same mother. A simple question, such as what species laid this egg case, may require DNA from a capsule collected thousands of meters down. The machines become part of the biology because they determine what can be seen. A 4K camera can reveal embryos, scars, arm positions, capsule texture, and subtle movement. A manipulator can collect a case without crushing it. A temperature probe can measure the warm seep that changes development. A shipboard lab can preserve tissue before it degrades. In the footage, the animal and the machine often share the frame. A robotic arm next to a palm-sized skate case. A camera housing reflected in particles. Thrusters stirring sediment near an octopus den. A suction sampler approaching a golden dome. The human presence is artificial, but the behavior it records is not. Researchers must also account for how animals respond to the machine. Lights can attract, repel, or alter behavior. Thrusters can disturb sediment. Noise and movement can change what an animal does. A brooding mother may hold position because she cannot flee, not because the vehicle is harmless. Observing without interfering is an ideal approached carefully, never perfectly. Despite these limits, ROV observations have transformed the subject. They have shown squid brooding eggs, octopuses nesting at thermal springs, skate egg cases near vents, cirrate eggs on corals, and many other reproductive moments that could not be understood from dead specimens alone. Video does not replace anatomy or genetics, but it gives behavior back to animals once known mostly as bodies in nets. Behavior is what makes the deep sea feel inhabited. A preserved squid in a museum jar can show hooks, arms, mantle, beak, and reproductive organs. A living squid carrying eggs shows commitment, vulnerability, and motion. A skate egg case in a collection can show shape and species. A field of cases near a black smoker shows place. A coral fragment with a capsule can show attachment. Footage shows relationship. Relationship is the missing piece in many old deep-sea collections. For decades, animals came up from depth flattened, torn, pale, or swollen from pressure change. Soft tissues lost form. Colors changed. Stomachs everted. Eggs separated from adults. The ocean was studied through fragments, and scientists did remarkable work with them. But now the fragments can sometimes be placed back into scenes. Scenes matter because reproduction is spatial. The difference between one rock and another can decide whether an egg receives enough flow. The difference between one side of a vent field and another can decide whether warmth helps or kills. The difference between a coral branch and sediment can decide whether a capsule is smothered. Eggs make microhabitat visible. A microhabitat is small from a human perspective, but for an embryo it can be the whole world. The inside of a capsule, the water moving through it, the surface it is attached to, the temperature around it, the predators able to reach it, the microbes growing on it, the mother's body if she is present. Nothing beyond that matters until hatching. After hatching, the scale changes instantly. A young skate emerges into benthic life. A young octopus leaves the protection of a mother and crevice. A squid hatchling enters the water column. A larva from a vent animal enters currents that may carry it away. The boundary opens, and the animal must begin sensing, moving, feeding, and avoiding being eaten. Every egg is therefore a temporary world with an expiration date. The deep sea complicates that expiration date. In cold habitats, development may be so slow that the temporary world lasts for years. This extended development can produce more capable young, but it ties success to stability. A case must remain intact. The environment must remain suitable. The parent, if brooding, must survive long enough. The surface above may change, but the embryo below continues at its own pace. Human activity can now change deep sea conditions faster than many of these life histories are built to absorb. Bottom contact fishing can damage corals and sponges that serve as egg attachment sites. Warming can alter oxygen and temperature regimes. Deoxygenation can compress habitable layers. Mining interest in vent and nodule regions raises questions about nurseries that may not yet be mapped. The reproductive stage is often the least resilient to disturbance. This does not mean every deep sea egg is currently threatened by the same activity, or that every nursery is known to be at risk. The evidence varies by region and species, but it does mean that reproductive habitats should be treated as more than background. If a place supports eggs, brooding, or repeated laying, its importance may be greater than a simple count of adult animals would show. A broken coral may be a broken nursery. A buried vent margin may be a lost incubator. A disturbed slope may be more than sediment in motion. Strange eggs ask us to look at the deep sea through the next generation instead of only through the adult body. That shift changes the whole image. A seamount becomes a gathering place. A vent becomes a source of developmental heat. A coral garden becomes a set of raised platforms. The open midwater becomes a space where a mother carries her offspring through darkness. The animals themselves do not know the ocean as a single global system. They know local conditions. Flow along a canyon. Temperature at a spring. Texture of rock. Chemical traces in water. The presence of mates. The pressure of predators. Evolution works through those local details, and eggs record them. This is why scientists often resist broad statements after one discovery. A skate using vent heat at Galapagos does not prove all deep-sea skates do the same. An octopus nursery at Davidson Seamount does not explain all deep-sea octopus reproduction. A brooding squid in Monterey Bay or the Gulf of California does not make brooding normal for all squid. Each discovery opens a category of possibility. Possibility is different from certainty, and in the deep sea that difference is worth preserving. But even limited discoveries can alter the imagination of a habitat. Before the ROV footage, a vent field might be thought of mainly as a place of tube worms, shrimp, mussels, microbes, chimneys, and chemistry. After the skate eggs, it is also a place where a fish from the wider deep sea may leave its young to develop in borrowed volcanic warmth. Before the long-term study, an octopus aggregation might look like a strange concentration of adults. After the hatching data, it becomes an incubator with measurable consequences. Before brooding squid were filmed, a squid egg mass was expected to be abandoned. After the footage, a mother in the midwater could no longer be assumed to be alone. That is how the deep sea changes. Not all at once, but by removing assumptions one image at a time. Eggs also help connect deep-sea animals to their surface relatives without making them identical. A skate case washed onto a beach and a skate case near a black smoker are both built around the same broad reproductive mode, but their environments differ profoundly. A shallow octopus tending eggs in a den and a pearl octopus nesting near abyssal thermal springs share maternal care, but the timescales and temperatures differ. A squid releasing eggs and a squid brooding eggs belong to the same larger lineage, but their strategies diverge. Evolution does not invent from nothing each time. It modifies existing possibilities. A tough egg case becomes useful in deeper water. Maternal care becomes extended in cold environments. Brooding in arms becomes a way to keep embryos oxygenated in the water column. Attachment to coral becomes a way to lift eggs into current. The deep sea reshapes old tools. Sometimes the reshaping produces forms that look almost designed for mystery. Horned purses, golden domes, pearled octopus fields, gelatinous sheets, capsules ruptured at one end. But their meaning is not in how mysterious they appear to us. Their meaning is in what they do. The case holds, the mother guards, the vent warms, the current breathes, the capsule opens. Deep-sea reproduction can also be inferred from anatomy when eggs are not visible. Enlarged ovaries, spermatophores, brooding structures, egg size, yolk content, and larval forms all provide clues. In some cephalopods, the presence of spermatangia on females can reveal mating. In fish, gonad condition can indicate spawning readiness. In invertebrates, egg capsules or larvae can connect adults to developmental stages. But inference has limits. An animal with mature eggs in its body does not show where it will lay them. A larva in a plankton sample may not show where it came from. A damaged adult in a trawl may not reveal whether it guarded eggs, released them, or carried them. Direct observation remains rare and therefore powerful. The danger is that rare footage can become overinterpreted because it is beautiful. A single video can travel farther than the paper that explains it. The image becomes simplified. A possible new species becomes a confirmed monster. A suspected egg becomes an alien object. A nursery becomes a miracle rather than a habitat with measurable temperature, depth, species, and risk. Good narration should resist that simplification. It can allow wonder without giving up caution. It can say that a squid may be an undescribed species without declaring it as fact. It can say that vent warmth likely accelerates development without pretending every embryo was measured from laying to hatching. It can say that the golden orb looked like a possible egg before laboratory evidence showed otherwise. The deep sea is strange enough without being exaggerated. The pressure, the darkness, the cold, the slow fall of food, the chemical islands, the rare nurseries, the mothers that die after care, the embryos waiting years inside cases, the animals seen once in decades of diving. All of this is already larger than invention, The deepest parts of the ocean, including trenches, add further questions. Hadal environments below 6,000 meters are shaped by high pressure, low temperature, and the funneling of organic matter into trench axes. Animals there include amphipods, holothurians, microbes, fishes in some trenches, and other specialized life. Their reproductive strategies are less visually documented than those of many shallower deep-sea animals, but eggs and embryos must still solve the same core problems. It would be easy to imagine trench eggs as more mysterious simply because the habitat is deeper. But depth alone should not become a substitute for evidence. In some cases, hadal animals may have life histories similar in principle to relatives at lesser depths, modified by pressure, temperature, food supply, and isolation. In other cases, unique adaptations may exist. The honest answer is that many details remain poorly known. The camera has visited trenches, but not often enough to make their nurseries familiar. A lander may record scavengers. A submersible may see sediment and animals. Samples may bring back amphipods or microbes. But the egg stage can still pass unseen. The same is true of submarine canyons. Canyons cut into continental margins and can channel organic matter from shallow water into depth. They can be rich, dynamic habitats with currents, slopes, sediment flows, corals, sponges, fishes, and invertebrates. They may also provide attachment sites and food pulses that influence reproduction. Yet many canyon egg habitats remain only partly described. Seamounts are especially important because they alter currents and concentrate life. Water moving over a seamount can increase mixing and bring food to suspension feeders. Hard surfaces allow corals and sponges to grow. Predators and prey gather. The octopus garden at Davidson Seamount is one reproductive example, but seamounts around the world may host many others that have not been studied with the same persistence. Seamounts are also vulnerable. Their summits and slopes can be targeted by bottom trawling. Corals that took centuries to grow can be broken quickly. If eggs are attached to those structures, the damage affects more than adults. It removes the architecture of reproduction. This is why documenting eggs is not merely a matter of curiosity. It can inform protection. A place that looks like rocky habitat may be a repeated nursery. A cluster of cases may indicate long-term use. A brooding aggregation may mark a site that a population depends on. Without reproductive evidence, management may underestimate the consequences of disturbance. In the deep sea, the future is often attached to something small. Some reproductive structures are found only after they wash ashore or rise into shallower water. Empty shark or skate egg cases on beaches have helped citizen scientists and researchers map species distributions in coastal regions. Deep sea cases rarely offer that convenience, but the principle is similar. The case persists after the animal leaves. It becomes a clue. Identification keys for egg cases can use shape, size, horns, keels, tendrils, texture, and other features. For some groups, these traits can point to species. But deep-sea diversity complicates the task because many species are rare, poorly sampled, or not represented by complete developmental material. DNA can help, but only if tissue remains and contamination is managed. A single empty capsule may not tell a full story. It may not reveal whether it hatched successfully, when it was laid, or how far it moved. But in numbers, patterns emerge. Repeated finds at the same depth, on the same substrate, near the same environmental feature, can point toward nursery behavior. The deep sea often needs patterns because individual moments are so easily ambiguous. Pattern is what turns the strange into the known. The process can begin with something as simple as a pilot saying that the seafloor looks different ahead. A cluster appears. The camera zooms. Scientists discuss whether the objects are living animals, cases, sponges, or debris. The vehicle moves closer. A laser scale puts size into the frame. The manipulator reaches. The ship above waits on a cable that disappears into black water. The cable matters. It is the physical line between the surface world and the nursery below. Through it pass power, data, commands, and the fragile permission to look. If weather turns, if a thruster fails, if the cable tangles, the observation ends. The eggs remain. The ship leaves. Many deep-sea discoveries are like that. They are brief intersections between human equipment and biological processes that continue before and after the encounter. The skate embryo keeps developing after the RUV departs. The octopus mother keeps breathing water across her brood. The squid keeps drifting. The coral-attached capsule keeps swelling. The story does not wait for the camera. This makes narration difficult in a useful way. The voiceover cannot pretend to see everything. It must move between what was observed, what is known from related species, what the evidence suggests, and what remains uncertain. It must let the animal continue beyond the frame. A good deep-sea image is always incomplete. The incompleteness is not failure. It is a reminder of scale. Every RUV dive samples a tiny portion of a seafloor, larger than all terrestrial habitats, combined when considered across depth and area. Every midwater transect cuts a narrow path through a living volume. Every specimen is one body from a population whose boundaries may be unknown. For eggs, this means scientists often work with fragments of life histories. They may know the adult, but not the egg but not the larva but not the nursery but not the migration path. Connecting these stages requires different tools and often different specialists. Reproductive biology is a puzzle spread across depth, time, and methods. Environmental DNA may help in some cases. Water samples can contain traces of genetic material shed by animals, including cells, mucus, waste, and perhaps material associated with reproduction. eDNA can indicate that a species is present or has recently been present, even when cameras do not see it. But eDNA does not usually show behavior by itself. It can point to where to look. If eDNA from a skate appears near a vent nursery, it supports presence, but the egg cases and video show reproductive use. If DNA from the golden orb matched Relicanthus, it solved identity, but the in situ images helped connect the structure to a base attached to rock. Genetic tools are powerful, but they are strongest when paired with visual and ecological context. The future of deep-sea egg discovery will likely come from this pairing: better cameras, longer deployments, environmental sensors, robotic sampling, genetic analysis, and careful return visits. Not one tool replacing another, but many tools narrowing the distance between image and understanding. There may also be discoveries in old collections. A museum jar can hold a specimen collected decades ago, waiting for a question that did not exist when it was preserved. Egg capsules, brooding structures, or reproductive tissues may be re-examined with modern imaging or DNA. The deep sea is not only explored by going down, sometimes it is explored by looking again at what was already brought up. This was true for many groups of animals. Specimens collected by trawls, dredges, and expeditions become reference points for later work. When a new RUV video shows an unknown squid with eggs, scientists can compare its body shape with preserved relatives. When an egg case is collected, it can be matched to known species or reveal that the known record is incomplete. Collections hold context of a different kind, but collections also carry the marks of damage. Deep-sea animals brought up through pressure, and temperature changes may not resemble their living form. Soft bodies tear. Gelatinous tissues collapse. Colors fade. Egg masses can separate. This is why the living footage has such force. It restores posture, placement, and motion to bodies that preservation can flatten. A brooding squid in a jar would be a strange specimen. A brooding squid in the water is a life history. There is another kind of strangeness in the fact that some deep-sea eggs are found in places that seem severe even by deep-sea standards. Black smoker fields. Abyssal thermal springs. Coral colonies in cold currents. Midwater layers patrolled by predators. The beginning of life does not always happen in the softest places. Sometimes it happens where one danger is accepted because another is reduced. The skate accepts chemical and thermal complexity to shorten development. The octopus accepts crowding and the presence of scavengers to use warmth. The squid accepts reduced mobility to keep eggs oxygenated and protected. The cirrate octopod uses exposed coral branches to keep eggs in flow, even though exposure may make them visible. These are trade-offs, not victories. The deep sea is built from trade-offs. Even the choice between many small eggs and few large eggs is a trade-off between probability and preparation. In productive shallow waters, large numbers of small eggs may work because larvae can feed in plankton-rich environments. In deep or food-poor waters, larger eggs with more yolk can produce young better prepared for scarcity. But this pattern is not universal. Life histories vary, and each species must be considered on its own evidence. General rules help, but they can hide exceptions. Some deep-sea animals produce many small eggs. Some shallow animals produce fewer larger eggs. Temperature, predation, larval feeding mode, adult size, oxygen, and evolutionary lineage all matter. The deep sea pushes certain pressures strongly, but it does not force every lineage into the same solution. That diversity is visible even within cephalopods. An octopus mother guarding eggs in a crevice, a cirrate attaching single eggs to coral, a gonatus squid carrying thousands, a possible undescribed gonatid carrying a few dozen giant eggs, and a bathyteuthis holding a gelatinous sheet all belong within a broad cephalopod story, but none can stand for all the others. Cephalopods are often intelligent, mobile, and behaviorally complex, but their reproductive lives can be brutally final. Many are semelparous, reproducing once and dying afterward. This pattern makes brooding especially intense. The parent's final energy is focused on one reproductive event. In deep water, where embryonic development can be long, that event may last a large part of the adult's remaining life. The death of the mother is not a failure of care. It is the endpoint of the strategy. At the octopus garden, dead adults become food for scavengers. In the midwater, a brooding squid may be eaten before or after hatching. The parent's body returns to the food web. The eggs, if successful, move forward. The system closes without sentiment. But the footage makes it hard not to feel the stillness of it. A mother octopus under a rock ledge at 3,200 meters is not aware of the research ship, the paper, the live stream, or the human fascination with her brood. She is doing what her lineage has made possible. Yet the image crosses an enormous distance. It brings a hidden reproductive act into a room full of screens, then into the minds of people who may never see the ocean beyond the surface. That translation can be valuable if it remains accurate. The public attention given to the golden orb showed how quickly a deep-sea object can become a story. The later identification showed why patience matters. The orb was not an egg, but its journey through speculation and analysis made the public watch the process of uncertainty being resolved. In that sense, false eggs can teach us about real eggs. They show that appearance is only the beginning. They show the need for samples and specialists. They show that the deep sea contains structures whose function may not be obvious when separated from the animal that made them. They show that the first question can be wrong and still lead toward knowledge. The ocean is not embarrassed by our wrong guesses. What matters is whether the guesses remain flexible. The deep sea animals that produce eggs also live within food webs shaped by other reproductive events. Lanternfish spawn. Copepods reproduce. Jellyfish release young. Crustaceans carry eggs. Amphipods brood. Worms release larvae. Corals spawn or brood depending on species. The strange eggs that attract cameras are only a visible part of a much larger reproductive field. Most of that field is small. Planktonic eggs and larvae can be nearly transparent. Many are identified only under microscopes or through DNA. Some are collected in nets that sample depth layers. Others are eaten before they are ever recorded. The deep sea's future often moves as specks in water, far below the threshold of ordinary vision. Yet those specks sustain the larger animals that later appear in the beam. A fish hovering over sediment began as an egg. A squid with black eyes began as an embryo inside a mass. A skate gliding near the bottom began inside a leathery case. A giant anemone attached to rock began as a larva that found a surface. Every adult deep-sea animal is evidence that some egg, larva, or embryo once survived. The adult body is the egg's afterlife. That survival is not guaranteed, and it may become harder as ocean conditions change. Deep waters are warming in many regions, though slowly compared with the surface. Oxygen levels are changing. Food supply from the surface can shift with climate and productivity. Human activity reaches farther down through fishing, mining exploration, pollution, and noise. Life histories shaped around slow development may not respond quickly. The exact effects will differ. Some species may tolerate change. Some may shift depth. Some may reproduce differently if conditions allow it. Some may be constrained by habitat, especially species tied to vents, seeps, seamounts, coral gardens, or oxygen boundaries. The danger is not one simple global outcome, but many local disruptions to processes we have only begun to document. Protecting deep-sea reproduction requires knowing where it happens. That means mapping habitats, identifying nurseries, limiting damage to vulnerable structures, and treating rare observations as reasons to look more closely rather than as isolated curiosities. It also means preserving specimens and data so that future researchers can ask better questions than we can now. A nursery discovered today may become a baseline tomorrow. In the future, a robot may return to the same vent field and find new layers of skate cases. It may return to the octopus garden and record another generation. It may cross a coral garden and see capsules where none were noticed before. It may follow a brooding squid long enough to watch water move through the egg mass. It may collect a strange object that looks like an egg and prove that it is something else entirely. Each return reduces the distance between event and understanding. The first visit finds the shape. The second finds the pattern. The third measures change. The lab gives identity. The long record gives life history. Deep sea science advances not only by going deeper, but by returning carefully to what once seemed only strange. The eggs wait through all of this without knowing they are evidence. A skate embryo inside its case does not know it lies near a black smoker. It responds to temperature, oxygen, development, and its own body. An octopus embryo does not know it belongs to the largest known aggregation of octopuses. It develops under the mother's arms. A squid embryo in a gelatinous sheet does not know it is changing assumptions about squid behavior. It grows while water moves past it. Human meaning comes later. Still, there is something powerful in the alignment between small beginnings and enormous places. A single egg case against a volcanic landscape. A capsule on a coral branch in an ocean basin. A brood under a mother at the base of a seamount. A sheet of eggs suspended in a water column kilometers deep. The scale difference is almost impossible to hold in the mind, but the camera holds it for a second. That second is enough to show that the deep sea is not only a place of survival, it is a place of inheritance. The animals there are not merely enduring pressure and darkness. They are finding ways to pass through them. The phrase "strange eggs" can therefore be read two ways. It can mean objects that look unfamiliar, and there are many of those. But it can also mean reproductive strategies that seem strange because they are shaped by conditions outside normal human experience. The second meaning is more important. The egg is strange because the world around it is strange to us. To the animal, the strategy is not strange. It is available, tested, and inherited. A skate does not choose volcanic heat in a human sense. A population persists if laying near warmth improves survival enough across generations. An octopus does not calculate the thermal advantage of a spring. Her behavior carries the history of those that hatched and returned. Evolution is not intention, but it can leave patterns that look almost deliberate. The pattern is what the ROV sees. The pattern can also disappear. If a vent stops flowing, a warm nursery may cool. If a coral garden is damaged, attachment sites may vanish. If currents shift, larvae may disperse differently. If oxygen declines, developmental success may change. Reproductive habitats are dynamic, and deep-sea animals must live within that dynamism. Some changes are natural. Some are increasingly human-driven. The challenge is that we may not notice loss at the egg stage. Adults may still be seen for a while. A population can carry the memory of past reproduction in its adult bodies even after current recruitment declines. By the time fewer young appear, the cause may be difficult to trace. Slow development delays both life and warning. This is why eggs deserve attention before they become absence. A strange egg found in the deep sea is not only an answer to what lives there, it is a question about what will live there next. There is another reason deep-sea eggs can feel unfamiliar. Many are not arranged in the neat forms that human eyes expect. They may not sit in a nest. They may not be round. They may not be white. They may not be fragile. They may not appear in groups that look like parenthood. A deep-sea egg can look like a seed pod, a wound, a brown leaf, a torn bag, a string of pearls, a sheet of glass, or a small growth on a living coral. Shape follows function, and function follows environment. A flat egg sheet can be held against a squid's arms. A long tubular mass can allow water to pass through it. A tough case can resist abrasion on lava. A capsule attached to coral can remain lifted above sediment. A gelatinous mass can protect many embryos while remaining nearly invisible in open water. None of these structures exists to be recognizable to us. Color is also unreliable. In the deep sea, red may appear black under natural light because red wavelengths disappear quickly with depth. Many animals that look bright red under ROV lights are effectively dark in their own environment. Egg cases may look brown, gold, black, cream, or translucent depending on lighting, age, preservation, and the material from which they are made. A color seen on a screen is not always the color an animal sees. This matters when footage spreads beyond the scientific team. A golden object becomes a golden egg. A pale cluster becomes alien spawn. A black case becomes something ominous. But under the ROV lights, the ocean is being artificially illuminated by a machine. The colors are real in one sense, but they are not the ordinary visual conditions of the habitat. The deep sea is not lit for human interpretation. The camera brings its own sunrise. The animals, meanwhile, respond to other cues. Chemical traces in water, texture under arms or fins, the movement of current across a surface. Temperature differences too small for a human hand to notice, but large enough to change embryonic development. Pressure that remains constant across a body because the body is built for it. Darkness that is not absence, but the normal state. The egg has no eyes for the scene in which we place it. It does not know that it is strange. Its world is the membrane, the yolk, the flow of oxygen, the chemical gradients, and the timing of its own development. It is not waiting to be understood. It is waiting to become capable of leaving. In many marine animals, the early embryo passes through stages that are recognizable across broad groups: cleavage, gastrulation, organ formation, movement, growth, and hatching. But the timing and outward appearance of these stages can differ enormously. A cephalopod embryo may develop visible eyes before hatching. A skate embryo gradually forms the flattened body and tail that will later leave the case. Larvae of many invertebrates may be tiny and planktonic, built for dispersal before settlement. The deep sea slows many of these processes because temperature affects biochemical reactions. Enzymes work differently in cold water. Cells divide more slowly. Embryos consume yolk more slowly. This can preserve energy, but it extends exposure. The result is one of the central tensions of deep-sea reproduction. Slow development can be efficient, but it can also be dangerous simply because it lasts. Warm seeps and vents become important because they change that tension. They do not make the water warm in the ordinary surface sense. They create pockets and flows where temperature is higher than the surrounding deep sea. For a developing embryo, a difference of a few degrees can matter. It can shift the clock of development without requiring the animal to leave the deep environment entirely. But warmer water can also increase oxygen demand. An embryo developing faster may need more oxygen. If the capsule cannot supply enough, speed becomes a problem. This is why the position of the egg relative to flow matters. Warmth without oxygen is not a nursery. Warmth with the right circulation can become one. A vent nursery is therefore not just heat. It is heat in motion. Current is often invisible in footage unless particles reveal it. Marine snow drifting sideways, a soft coral bending, a plume leaning away from a chimney, sediment lifting behind a thruster, or the slight movement of an egg strand can show water passing through a scene. For eggs, that invisible movement can be as important as the rock itself. It delivers oxygen and carries away waste. On a flat, muddy bottom, flow near the sediment surface can be weak. Boundary layers form where water movement slows. Eggs buried or lying too close to stagnant sediment may face reduced oxygen. Raised structures change this. Corals, sponges, rocks, chimneys, and ledges lift eggs into better exchange. The physical architecture of the seafloor becomes reproductive infrastructure. This is one reason cold water coral reefs and sponge grounds are so important in the deep sea. They are not only homes for adult animals, they are three-dimensional habitats in a world that can otherwise be flat for long distances. Small fish shelter there. Crustaceans feed there. Brittle stars climb there. Eggs can be attached there. Larvae can settle there. The structure creates possibilities. A destroyed structure cannot immediately be replaced. Some deep-sea corals grow slowly. Some live for centuries. A trawl pass can break branches in minutes that took generations to build. If those branches serve as egg attachment sites, then the loss is both present and future. It removes animals now, and removes surfaces where future animals might begin. The threat is not always visible in the same way. Broken coral is obvious, missing eggs are not. A field may look barren because it is naturally sparse, or because its reproductive structures were removed before they were documented. Without baseline observations, absence is hard to interpret. This is one of the hardest problems in deep-sea conservation. You cannot protect a nursery you have not yet recognized. There are hints that some animals return to reproductive sites over long periods. Layers of skate cases suggest repeated laying. Octopus aggregations persist where thermal conditions remain favorable. Fish spawning grounds in other marine systems can be used across generations. In the deep sea, repeated use may be especially important because suitable habitat can be rare and widely separated. A site can be suitable because of temperature, chemistry, flow, substrate, depth, predator conditions, or a combination of these. For the Pacific white skate near the Galapagos, the vent field appears to provide warmth. For pearl octopuses at Davidson Seamount, warm springs speed development. For cirrate eggs on corals, elevated attachment may provide flow and stability. For brooding squid, the suitable site is not a site at all, but a depth range through which the mother moves. This variety makes it difficult to search for deep-sea nurseries with one method. A sonar map can reveal seamounts, ridges, canyons, and vent chimneys, but not transparent egg sheets. A camera transect can reveal visible cases, but not embryos hidden in sediment. A plankton net can collect larvae, but not always connect them to adult habitat. A temperature anomaly can suggest a possible incubator, but only if animals use it. The search is slow because the target changes form. Sometimes the nursery is a crowd. Sometimes it is a patch. Sometimes it is a single mother. Sometimes it is a drifting mass that may never pass through a camera beam again. This is why the deep sea can seem to offer discoveries as accidents. The rove was there at the right time. The camera turned at the right moment. The animal did not flee. The egg case had not yet hatched. The mother had not yet died. The current had not yet carried the mass away. But behind the accident is preparation. Ships are planned. Maps are made. Vehicles are built. Pilots train. Taxonomists wait for footage and samples. Oceanographers measure water. Engineers design pressure housings that do not fail in darkness. A discovery that looks sudden may rest on years of ordinary work. The same is true after the dive. Samples must be preserved correctly. Metadata must be stored. Video must be reviewed. Specialists must be consulted. If DNA is used, contamination must be controlled and sequences compared. If developmental timing is estimated, repeated observations must be matched carefully. The strange egg becomes knowledge only after the slow work begins. The deep sea is patient, and it forces patience on anyone trying to understand it. One of the most interesting questions is whether some deep-sea animals choose nursery sites actively, or whether their behavior is simpler than that word suggests. A skate laying eggs near a vent may detect temperature, chemistry, substrate, or some combination of cues. An octopus may be attracted to warm cracks, the presence of other octopuses, or both. A squid may move to a depth that balances oxygen, temperature, and predator risk while brooding. Choice does not need to mean conscious planning. It can mean inherited behavior guided by sensory cues. Animals can behave in ways that look strategic because selection has favored individuals that respond to the right signals. A female that lays eggs in a slightly warmer, better oxygenated place may leave more surviving offspring. Over generations, that response can become a pattern. This is how nurseries can form without maps, names, or memory in the human sense. Adults respond to conditions. Offspring survive at different rates. The next generation carries the tendencies that worked. The result is repeated use of places that meet the requirements of development. From the camera's perspective, it looks like purpose written into the seafloor. The purpose, however, is distributed across generations. No single skate needs to understand volcanic geology. No octopus needs to know the age of Davidson's seamount. No squid needs to describe the oxygen structure of the water column. Their bodies and behaviors are already tuned to parts of those realities. The sensory world of deep-sea reproduction is still not fully known. Chemical sensing is likely important for many animals. Mechanoreception, the detection of water movement and vibration, may guide behavior. Some animals may detect temperature gradients. Others may use touch to evaluate substrate. Bioluminescent signals may help mates find each other in open water. The egg stage begins after these sensory problems have been solved, but it depends on them. Before an egg appears, two adults or two sets of gametes must meet in an ocean that separates everything. That meeting can be the hardest hidden event. The egg is visible evidence of a successful encounter that may have been even harder to observe than the egg itself. Every egg case is a record of a meeting the camera probably missed. In some deep-sea footage, scientists notice reproductive clues only later. A structure on an arm. Swelling in a body. Eggs tucked beneath an animal. A capsule attached to a coral. During a live dive, attention is divided among navigation, sampling, species identification, terrain, and safety. Only after review does the reproductive meaning become clear. This delayed recognition is common in science. The first viewing captures data. The second viewing sees pattern. The third, with specialists involved, may see behavior. A frame that seemed ordinary during the dive can become important months later when someone notices the small pale objects under an arm or the ruptured edge of a capsule. High-resolution video has made this possible. Older deep-sea footage can be grainy, dim, or limited by camera technology. Modern systems can show fine textures, small embryos, individual suckers, scars on octopus skin, and subtle flow through egg masses. Better imagery does not remove uncertainty, but it gives uncertainty more detail to work with. Detail is a kind of mercy in the deep sea. It lets us see that a mother octopus is not just a pale shape. She has skin texture, arm posture, scars, eyes, and a brood beneath her. It lets us see that a squid egg sheet contains individual capsules. It lets us see that a skate case has horns and seams. It lets us see that a coral-attached capsule has ruptured in a way that may be part of hatching. Yet even the best image is a surface. It does not show metabolism directly. It does not show genetic identity unless sampled. It does not show the entire developmental history. It does not show how many embryos fail inside similar structures nearby. The visible is essential, but not complete. Scientists often combine observations with models. Developmental rates can be estimated from temperature and related species. Current models can suggest larval dispersal pathways. Habitat models can predict where suitable nurseries might occur. Population models can explore how long incubation affects vulnerability. These tools are useful, but they depend on assumptions that must be tested. Deep sea eggs are especially challenging for models because small changes can matter. A temperature difference of 1 or 2 degrees may alter development. A current shift may change oxygen delivery. A predator aggregation may increase mortality. A maternal behavior observed in one species may not apply to another. The model can guide the search, but the ocean decides the result. The ocean is always more specific than the model. There is also the question of sound and vibration. The deep sea is not silent. It carries whale calls, seismic activity, rain and wave noise from above, ice sounds in polar regions, ship noise, biological movement, and the low mechanical presence of human activity. How eggs or brooding animals respond to sound is not well known for many species, but adults may be sensitive to disturbance in ways that influence reproductive behavior. It would be inaccurate to claim that every deep-sea nursery is threatened by noise in the same way. The evidence is uneven. But it is reasonable to say that reproductive behavior can be sensitive to disturbance in many animals, and that the deep ocean is increasingly affected by human sound. As with other impacts, the egg stage reminds us to ask quieter questions before assuming no effect. The deep sea used to be protected partly by distance. That protection is shrinking. Fishing gear reaches deep slopes. Mining vehicles are tested for abyssal plains and vent regions. Cables cross basins. Plastics and chemicals arrive through currents and sinking debris. Climate-driven changes move through water masses. The eggs remain small, but the forces around them are growing larger. A slow life history can make recovery slow. If an animal takes years to develop inside an egg and years more to mature, then a damaged population may not rebound quickly. If a nursery site is lost, adults may not immediately find an equivalent place. If the habitat is rare, replacement may not exist nearby. Reproduction sets the pace of resilience. The future of a deep-sea animal is limited by how often it can begin again. Some species may be more resilient because they produce many offspring, disperse widely, mature quickly, or use common habitats. Others may be less resilient because they produce fewer eggs, develop slowly, depend on specific substrates, or gather at predictable nurseries. Without knowing reproduction, it is difficult to know vulnerability. This is why strange eggs have scientific value beyond their appearance. They reveal fecundity, investment, development, habitat use, parental care, and sometimes the location of critical life stages. They help translate an adult species from a point on a map into a life cycle. A life cycle is more fragile than a body. The body can move away from a disturbance. The egg often cannot. The adult can search for food. The embryo depends on stored energy. The adult can avoid some predators. The egg waits behind a case or under a mother's arms. The adult can sense changing conditions. The embryo experiences them through a membrane. This immobility is not total. Some eggs drift. Some mothers carry them. Some capsules may be moved by currents or animals, but compared with adults, many embryos have limited control over where they are. The laying site, brooding behavior, or buoyancy of the egg mass determines much of their early fate. The location of an egg is therefore an extension of the parent's ecology. A parent that selects a site well can increase offspring survival. A parent that releases eggs into a favorable current can increase dispersal. A mother that carries eggs can keep them in conditions she can tolerate, though perhaps at great cost. Reproduction links adult movement to embryonic environment. This link is beautifully visible in brooding squid. The mother is not only protecting eggs from being eaten, she is choosing, by every movement of her body, the water the embryos experience. Depth, temperature, oxygen, current, and predator field all travel through her behavior. The egg mass has no fins of its own, but it moves, because she does. If she weakens, the egg mass weakens with her. If she is eaten, the embryos may be eaten too. If she cannot ventilate them, oxygen may fail. Maternal care concentrates survival and risk into one body. It is both protection and dependence. On the screen, this can look like stillness. In reality, it is work. Octopus mothers also work while appearing still. They move water with arms and mantle, clean eggs, guard them from small predators, and maintain position. In shallow species, this has been observed in detail. In deep-sea species, the general pattern is supported by footage and related biology, but many specifics remain less documented. The deep mothers are seen less often, and their brooding periods can be far longer. The longest known brooding periods in octopuses are extraordinary. A deep-sea octopus observed off California was reported guarding eggs for more than 4 years, one of the longest brooding periods known for any animal. That example is not the octopus garden species, and it should not be applied to every deep-sea octopus. But it shows how extreme cold water maternal care can become. 4 years beside a clutch of eggs is not a dramatic chase or a sudden battle. It is endurance beyond ordinary imagination. The mother remains as her body declines. The embryos develop slowly in cold water. The seafloor changes in small ways around them. The world above goes through seasons that never reach them as light, but may reach them as food falling through water. In the deep sea, waiting can be the main event. The eggs found there make waiting visible. They are objects in time. A skate case near a vent is not only a structure, but a multi-year process. An octopus brood is not only a cluster, but a long maternal interval. A squid egg mass is not only a sheet, but months of reduced movement. A coral-attached capsule is not only a spot on a branch, but a developmental sequence ending in rupture and escape. If the script lingers on them, it should linger for that reason, not to make them mystical, but to respect the timescales they represent. A quick visual may be all the footage gives us. The biology beneath it may last far longer than the shot. The shot is brief. The egg is not. That difference may be the deepest part of the story. Human exploration enters the deep sea in flashes. A dive, a sample, a cruise, a publication, a clip. Reproduction there may unfold over years, across sites, through currents, from adults that gather and die to young that disperse unseen. The camera arrives as a visitor to a process already underway. This is why a final answer should never sound too final. The golden orb has been identified, but other objects will appear. The skate nursery is described, but other nurseries may exist. The octopus garden is monitored, but other thermal spring nurseries may remain unknown. Brooding squids have been recorded, but the full diversity of squid reproductive care is still incomplete. Science does not close the deep sea. It makes parts of it clearer. And in the cleared parts, the eggs remain strange enough. There is one more kind of egg that rarely becomes a headline because it can pass through the sea almost invisibly. In the open water, some eggs are carried inside gelatinous masses that are mostly clear. They can be large enough to surprise a diver and still difficult to see from a distance. They have no hard edges, no nest, no rock, no mother holding them in place. They are temporary architecture made of mucus, water, and embryos. In shallower seas, some squid egg masses have been found as large drifting forms, sometimes compared to balloons or transparent sacs. Many are short-lived, and the embryos inside may hatch quickly compared with deep-sea embryos. In deeper water, similar structures may be produced and lost from view without ever being filmed. A gelatinous mass can exist in the water column, move with currents, and disappear before a ship crosses its path. These masses remind us that not all eggs are meant to resist the seafloor. Some are meant to float, sink slowly, remain suspended, or drift through a layer where temperature and oxygen are suitable. Their protection is not leather or shell. It is spacing, transparency, chemistry, and the sheer difficulty predators may have in finding and consuming every embryo before development is complete. But gelatin is not invulnerability. Jellies, fishes, crustaceans, worms, and microbes can all interact with soft egg masses. Currents can stretch or tear them. Low oxygen can harm them. A mass that looks intact may contain embryos at different stages or embryos that have already failed. Without close imaging or collection, the condition of the young inside may remain unknown. The ocean can carry a nursery like smoke. In black water, such a mass might enter the edge of the lights for only seconds. The ROV pilot may be focused on a seafloor target below. The scientists may be discussing rocks, corals, or fish. A translucent structure can pass above the frame, visible only because marine snow bends around it or because the light catches the embryos suspended inside. Then, it is gone. The deep midwater is perhaps the hardest place to understand eggs because there is no fixed background. On the seafloor, an egg case can be mapped to a coordinate. On a coral branch, it can be revisited if the site is found again. In the water column, the object is part of motion. It may rise, sink, rotate, stretch, fold, or drift miles away before anyone could return. Oceanographers divide the water column by depth, but animals experience it as gradients and layers. Temperature changes, oxygen changes, pressure changes, prey layers form, predators patrol, currents shear. A gelatinous egg mass or a brooding squid passes through these conditions, and the embryos experience them without ever touching the bottom. This is why vertical position can matter as much as geographic location. An egg at 800 meters and an egg at 1600 meters may be in very different worlds, even if they are above the same seafloor. Light, temperature, oxygen, predators, and food webs change with depth. For drifting eggs, depth is habitat. There is no nest, but there is still a place. Some larvae that hatch from deep-sea eggs may move upward into waters where food is more available, then descend later as they develop. Others may remain in deep layers or near the bottom. The movement can be active or assisted by buoyancy and currents. For many species, these early migrations remain poorly known because the larvae are small and often difficult to identify to species. A larva collected in a net may be assigned to a family, but not a species. A tiny cephalopod hatchling may not yet have the features used to identify adults. A fish larva may look nothing like the bottom-dwelling adult it will become. DNA barcoding can help connect stages, but reference databases must include the adult species, and many deep-sea animals remain incompletely sequenced. This means that the egg stage, larval stage, juvenile stage, and adult stage can be separated in the scientific record. One paper may describe adult anatomy, another may describe larvae from plankton, another may show eggs on a coral. Connecting them can take years. A life cycle can be known in pieces before it is known as a cycle. The animal lives continuously. Human knowledge does not. In the deep sea, even the word hatch can cover many different transitions. A skate leaves a case as a small version of the adult form. An octopus hatchling may crawl or swim away with a body already suited to predatory life. A squid hatchling enters the water column as a paralarva, equipped for a different scale of feeding and movement. A snail larva may enter plankton before settlement. The opening of the egg is not one event across all animals. What is shared is the crossing of a boundary. Inside, the embryo is protected and limited. Outside, it is free and exposed. The moment of hatching is both escape and danger. The egg has done its job by ceasing to hold. Many will be eaten almost immediately. Small hatchlings and larvae are prey to animals only slightly larger than themselves. A transparent body does not guarantee safety. A quick start does not guarantee food. The first hours or days after hatching may be as dangerous as the entire period inside the egg. Protection ends before competence is complete. This is why yolk reserves can matter after hatching. Some young animals emerge with remaining internal energy, allowing them to survive before their first successful feeding. Others must begin feeding quickly. Larger eggs often provide more yolk and produce more developed young, but again, this comes at the cost of producing fewer offspring. The arithmetic of reproduction is always present. If a mother produces 3,000 small eggs and only a few survive, the strategy can still succeed. If she produces 40 large eggs, each loss is proportionally greater, but each survivor may be better prepared. The Gulf of California squid with giant eggs makes this trade-off visible. The egg size itself becomes an ecological statement. A larger egg is a longer promise. There may be deep-sea eggs that humans have seen many times without recognizing them. A rounded capsule on a coral might be mistaken for part of the coral. A clutch under a rock might be hidden by the adult. A gelatinous sheet might be dismissed as damaged tissue. A case partly buried in sediment might look like debris. Recognition often depends on the right specialist watching the right footage with the right question in mind. This is why open data and archived video matter. A dive recorded for geology can later reveal biology. A survey of mineral resources can capture a nursery. A camera test can record rare behavior. When footage is stored and searchable, discoveries can be made long after the vehicle is back on deck. The deep sea record is full of such delayed value. A specimen collected for one reason becomes useful for another. A photograph taken for mapping becomes evidence of reproduction. A temperature reading collected during a dive becomes the key to understanding development. The ocean does not separate disciplines neatly. A biological answer may require geology, chemistry, engineering, taxonomy, and patience. Eggs sit exactly at that intersection. They are biological structures placed into physical environments, shaped by chemistry, constrained by temperature, dependent on flow, vulnerable to predators, and discoverable only through technology. To understand one strange egg, a scientist may need the seafloor map, the water properties, the species identity, the developmental biology, and the behavior of the parent. The small object asks a large set of questions. That is why the deep-sea egg can feel heavier than it looks. Not physically, but scientifically. It carries the unknown adult if the species is not identified. It carries the unknown habitat if the laying site is not understood. It carries the unknown future if hatching success is not known. It carries the limits of the dive that found it. The best response is not to make the egg more mysterious than it is. The best response is to let it be specific. A skate case near a black smoker. A pearl octopus brood at 3,200 meters. A Gonatus squid holding thousands of eggs from hooks on her arms. A Bathyteuthis sheet with hundreds of embryos. A cirrate egg on bubblegum coral. Specificity is what keeps wonder honest. Specificity also prevents the deep sea from becoming a single mood. The deep ocean contains quiet plains, steep canyons, active vents, cold seeps, brine pools, oxygen minimum zones, whale falls, coral gardens, seamounts, and open midwater. Eggs appear differently in each. A brine pool edge, for example, is a chemical boundary where hypersaline water can collect like a lake on the seafloor. It may be lethal to many animals, but surrounded by communities that exploit the gradient. It would be wrong to claim that brine pools are known primarily as egg nurseries without specific evidence. but they show the kind of sharp environmental boundary that matters in the deep sea. Animals may gather at edges, avoid interiors, feed on associated communities, or use nearby structures. Reproductive behavior in such places would need to be documented carefully before being described. The deep sea invites comparison, but it punishes assumption. Whale falls offer another example of temporary habitat. When a whale body reaches the seafloor, it creates a rich island of food that can support scavengers, enrichment opportunists, bone-eating worms, microbes, and sulfophilic communities over time. Eggs and larvae are part of that succession because animals feeding and living there must reproduce somewhere. But the whale fall itself changes as the carcass is consumed. Some animals at whale falls may release larvae that disperse to other organic falls or suitable habitats. Others may arrive as larvae from elsewhere. The carcass is not permanent, so reproductive timing and dispersal are crucial. As with vents and seeps, patchiness shapes life histories. The deep sea contains many islands that are not land: vents, seeps, carcasses, woodfalls, kelp falls, and hard substrates rising from sediment. Eggs tied to temporary habitats face a different problem from eggs tied to long-lived structures. If the habitat disappears quickly, offspring must disperse or mature fast enough to continue elsewhere. If the habitat is stable but rare, larvae must find it across distance. If the habitat is predictable but vulnerable, repeated use can become a liability under disturbance. There is no universal solution. Every nursery has an expiration date. Some are measured in years, some in centuries, some in the life of a mother. The more we learn, the more the deep sea appears not as a blank darkness, but as a mosaic of reproductive opportunities. Some are chemical, some are physical, some are behavioral, some are carried in bodies. Some are built from structures left by other animals. The egg is where these opportunities become visible, and because eggs are vulnerable, they reveal the deep sea at its least armored. An adult skate can swim. An adult octopus can move, hide, and hunt. A squid can jet away when not brooding. A coral can withdraw polyps. A snail can crawl. An embryo inside a case can only develop or fail. Its world is limited to the protection chosen before it had any agency. Agency is a difficult word for embryos, but vulnerability is not. Embryos depend on conditions. Development is conditional. Hatching is conditional. Survival after hatching is conditional. The deep sea is full of animals that seem independent as adults, but their life cycles begin in stages of profound dependence. The hidden ocean is built from dependent beginnings. One of the quiet lessons in all of this is that reproduction is not only about producing young, it is about placing young into a physical world. At the surface, that placement might be a nest on a beach, a patch of algae, the underside of a rock, or a floating mat. In the deep sea, it may be a vent margin, a coral branch, a crevice warmed by fluid, a sheet held in a squid's arms, or a depth layer moving through darkness. The egg is therefore a map point, even when it drifts. It records a choice made by an adult body or a lineage. Here, now, this temperature, this flow, this amount of protection, this number of offspring. Some of those choices are visible only after many examples are gathered. One case is a clue. A field of cases is a pattern. A pattern, repeated through time, becomes part of the species' natural history. Natural history can sound old-fashioned, but in the deep sea it is essential. Before a population can be modeled, protected, or understood, someone has to know where it lives, what it eats, how it moves, how it mates, where it puts its eggs, and what the young look like. For many deep-sea animals, even these basic facts are incomplete. That incompleteness is not empty. It is an invitation to look more closely. A future dive may find a skate laying an egg rather than only the egg case afterward. Another may record the first hatchling leaving a vent-warmed case. Another may watch a squid brood long enough to show how often she pumps water through the mass. Another may find that a familiar coral garden has been carrying octopod eggs all along. The discoveries ahead may not be louder than the ones already made. They may be smaller. A change in a capsule. A faint movement inside a case. A mother turning her arms. A hatchling leaving through a tear. A pattern in archived footage. A DNA match from a structure that looked anonymous on the day it was collected. The deep sea is often described by what humans cannot do there. We cannot walk across it easily. We cannot breathe there. We cannot remain there without machines. We cannot see far through it. But the animals are not living in a place of human inability. They are living in their own habitats, and eggs show that those habitats are complete enough to begin life, not just to sustain it after it has already begun. That distinction matters. A habitat that can support adults but not reproduction is not a lasting home. It is a feeding ground, a refuge, or a temporary range. A habitat where eggs develop, hatch, and send young into the population is part of continuity. When researchers find eggs, they find evidence that the deep sea is not only occupied, it is renewing itself. Renewal in the deep ocean can look like a small object on a dark rock. It can look like nothing happening. It can look like an octopus that barely moves, a squid that drifts too slowly to seem safe, a leathery case that has been sitting near a vent for years, a capsule on coral that seems broken until someone recognizes the mechanics of hatching. The drama is often hidden inside a timescale too slow for ordinary attention. To see it, the eye has to slow down. This is why the smallest finds can change the feeling of an entire descent. A camera may spend an hour crossing sediment, stones, and animals that keep their distance from the light. Then it finds one object that cannot move away, one case or capsule or brood held in place by the needs of development. The scene narrows. The ocean is still enormous, but attention gathers around something no larger than a hand, or a fingernail, or a cluster hidden beneath an arm. For a moment, the deep sea is not measured by kilometers. It is measured by whether oxygen can cross a membrane, whether warmth reaches a case, whether a mother can last, and whether the young inside will make it to the first breath of water beyond the egg. The same narrowing happens in the laboratory, though the light is different. A capsule that looked plain on the seafloor may be turned gently under a microscope. Its surface may show layers, pores, fibers, scars, or tiny points where it attached to another living thing. A fragment may be taken for DNA. The color may change under alcohol or preservation. A structure that seemed anonymous in the video begins to acquire relationships. It resembles a known egg case. Or it does not. It contains tissue. Or it is empty. It carries the genetic trace of a skate, a cephalopod, a cnidarian, a mollusk, or something that sends the researchers back through papers and museum drawers. The object becomes less mysterious by becoming more connected. That connection is the real reward of studying strange eggs. They connect adult to embryo, habitat to behavior, geology to development, and observation to evidence. A deep sea egg is never only itself. It belongs to a parent that had to find food, avoid predators, mature, mate, and reach the right place. It belongs to a habitat that had to provide the right combination of surface, water, chemistry, and time. It belongs to a future animal that may or may not survive long enough to be seen again. When scientists identify an egg, they are not just naming a structure. They are placing one hidden stage back into the larger movement of life below the surface. The evidence we have now comes from many places. The Gulf of Alaska gave us an object that looked like an egg but belonged to an anemone's base. The Galapagos vent field revealed skate egg cases using volcanic warmth. Davidson's seamount revealed a massive octopus nursery accelerated by thermal springs. Monterey Bay and the Gulf of California gave footage of squid mothers carrying eggs. The Central Pacific showed cirrate octopod eggs attached to corals. These examples do not exhaust the subject. they are entry points. The deep sea contains countless reproductive strategies in fishes, crustaceans, worms, mollusks, echinoderms, cnidarians, and microbes. Many will never become famous because they are too small, too common-looking, or too difficult to film, but they are part of the same hidden continuity. Life in the deep sea does not begin with a roar. It often begins as a capsule, a case, a sheet, a cluster, a cell, a small movement under a mother's arm. From the surface, the ocean can look like a single plane of moving light. Beneath it, the water deepens through blue, then dim blue, then black. Red disappears first. Pressure rises. Temperature drops. Food thins. The ship becomes a point above. The cable descends. The ROV lights switch on. Marine snow crosses the beam. Then, on rock or coral, or in the arms of an animal, the next generation appears. It is easy to miss because it does not look like power. It does not look like a predator, a giant, or a spectacle. It looks like something small trying to last long enough. That may be the most accurate image of the deep sea. Not emptiness, not monsters, not a world beyond life. A place where the beginning is slow, costly, and often hidden, but still persistent. The strange eggs found there remind us that every habitat has to solve reproduction, or it is only temporary. A vent community, a seamount slope, a coral garden, a midwater layer, an abyssal plain, a trench, all of them remain living worlds only if bodies can become new bodies. Eggs are the mechanism by which the deep sea continues. When the next unusual object appears on a monitor, the first question may again be simple. Is it an egg? The answer may be yes. It may be no. It may take a sample, a microscope, a genome, a comparison, and several years to know. But the question is worth asking, because in the deep sea, even the wrong question can point toward a life history we have not yet understood. The camera moves on. The object remains behind. Or the mother carries it out of frame. Or the case sits among rocks for another year. Above it, kilometers of water shift in darkness. Around it, animals feed, crawl, drift, wait, and search. Inside it, if it is truly an egg, a body is assembling itself one cell at a time. It has no idea that it has been found. It only has to hatch.